Hello everyone! So today we'll be covering embryology and specifically muscle development. This was meant to be both muscle development and limb development, and limb growth and bone development. However, the topics are quite big, and I think that in order to explain things succinctly, I've split into two parts. So I'll be doing muscle development today, and then I'll be doing next week limb development and limb budding and how bones grow and how they develop themselves. So let's move on to the contents. So the first thing is going to be a summary of the key steps in life before birth, basically a summary of what you have learned in phase 1. Secondly, we'll be talking about the mesoderm, how that differentiates. We'll be talking about things called somites, then how the somites transfer into the myotome. We'll of course talk about regulation of muscle development, innervation of the axial skeleton, and finally some development of the skeletal muscle, smooth muscle, and cardiac muscle, and the differences between the two. As always, we will have sample questions. This will involve a mix of questions that I've seen, that I've had in first year, and questions that have been modified in that regard. There'll be a lot of histology, a lot of diagrams, a lot of labeling. And of course, as always, we still have our question and answer time. So if you have any questions, do submit them either in the live chat, and as soon as they come up, and I will address them, or through the Google Doc, and I will go through them. Okay, let's begin. So life before birth, as I said, this is going to be a summary. If you guys want a full video on life before birth, the first 3 weeks, then let me know. But otherwise, we'll just go through this quite quickly. So the first 2 weeks, or week 1 to week 2, we have fertilization of the oocyte, we have cleavage, we have something called compaction, which forms our morula, and a blastocyst. So the product of all this from week 1 is a blastocyst. Week 2, we get implantation. So we have our bilaminar germ disc becomes the epiblast and hypoblast, and then the surrounding tissues, so the trophectoderm, they become the placenta, which eventually becomes placenta, and this is the syncytiotrophoblast and cytotrophoblast. Gastrulation, so this is week 3, so this is when we have 3 things. So week 2 bilaminar is 2, gastrulation is 3, so we have the ectoderm, mesoderm, endoderm, and we also have notochord and neural tube formation. Then we reach week 4 to 8, which is known as the embryonic period. We have the closure of the neural tube, we get 4 limb buds and 4 chambers of the heart. And this is also where the organs start to develop, so this is when you start making a proper embryo. And just the difference between the bilaminar and trilaminar discs. The bilaminar disc is an embryonic disc. You have the amnion, you have the yolk sac, hypoblast, and epiblast and hypoblast. In week 3, we have the 3 different layers. So trilaminar disc, so ectoderm, mesoderm, and endoderm. So the first we'll talk about mesoderm. So there's quite a lot to cover with the mesoderm. So this mesoderm differentiates into its distinct regions in day 17 to day 21. So this is a transverse section. So if you can imagine the embryo being like a tube and it's transverse, they cut. This is what we'd be seeing. So we have different types of mesoderms, and we have the paraxial mesoderm, we have the intermediate mesoderm, and the lateral plate mesoderm. Now all of these are important, but we'll be looking at the overall picture here, especially for muscle development. So this is what it looks like from a bird's eye view. So this would be the head, this would be the bottom, so the cranial, caudal, and we have something called a primitive streak. And this primitive streak is a result of the invaginations and the curvature that we witness in the neural tube and all the other places here. So as I said, paraxial mesoderm becomes the somites, intermediate mesoderm becomes the genitourinary tract, so this becomes into mesonephric ducts and metanephric ducts, so this becomes your kidneys. Lateral plates, this is your somatic and splanchnic layers, which are also known as the parietal, somatic parietal, and splanchnic visceral layer, and they form the lining of different organs. The paraxial mesoderm is also very important because it forms the cells and it moves sort of bilaterally and then cranially, which forms this primitive streak. So this paraxial mesoderm, its invagination is what causes this primitive streak. And it's adjacent to the notochord. So the notochord is this little black spot here. It's right next to that. And once again, it gives rise to the somites. So if you can think of the neural tube as the neural part, the mesoderm, the paraxial mesoderm starts forming first. And when it forms these tubes here. So this is a neural tube, this is the paraxial mesoderm, intermediate mesoderm, lateral plate mesoderm, and then the extraembryonic mesoderm, which is not really relevant to what we'll be discussing here, but it's just other mesoderm that's present. And this is a better way to notice this. We have the endoderm, ectoderm, for neural crest cells, and this is a good diagram to keep in mind because this is what forms, this is what you'll be asked about most commonly. As I said, we have the somatic and splanchnic, two layers, and in between we have something called a coulomb. So this, or the coelom, this is where you would have your insides of your gastrointestinal tract. So somatic, splanchnic, so parietal and visceral layers. Okay, moving on. So the mesoderm differentiates into many different things, and I think this is a very good summary of what happens. So we have the paraxial, intermediate, and lateral plate. So the lateral plate mesoderm, parietal layer becomes the bony wall and bones, whereas the visceral layer becomes the wall that lines the guts and the serous membranes. So it's the visceral layer of organs. So we will not be really looking at these parts, so the focus will not be on this, but we're focusing on this one here, because that's what we get for limb development. So we have somites. So somites, so all the axial skeleton, the body wall, and the limbs, all of this is derived from somites. And they undergo many different processes. They undergo epithelialization, which means that they become epithelialized, they become cells which start functioning within the tissue and start layering up. And the upper region of the cells, they develop into something called the dermamyotome, so which becomes the dermatome and the myotome. Dermatome is the dermis of the back, it's the connective tissue, whereas the myotome becomes the primaxial and abaxial muscles, which we will get into later. And then we have the lower region, which develops into the vertebrae and ribs. Now, the dorsomedial myoblast and lateral myoblast, which derive from the myotome, are part of the myotome, and they are the ones which become the muscles. And the relative location of those cells to the neural tube will determine if it's a primaxial or an abaxial muscle. I'll be showing you a diagram later on. But the summary of this sort of mesoderm differentiation is that everything is derived from somites. In order to get somites, you need paraxial mesoderm. And once the somites start epithelializing and undergoing transformations, and the cells which make up the mesoderm are committed to become somites, well, we get the dermatome and myotome in the upper region and lower region, vertebrae and ribs. So let's look at these somites. So this is what you would see at an embryo around day 20, and these little things are somites. Now the somites, the number of somites you see and how far down they go determines the age of the embryo, because they start forming from the head, so cranially, to caudally from day 20. So they often appear in pairs of 3, in 3 pairs a day, so they grow 3 more pairs until week 5. And this is how you can determine the age. This is a sort of cross-section of the somites here. So here we have the neural tube, which is closing up, notochord, and the mesoderm, paraxial mesoderm, which is close to the neural tube, which is developing into a somite. Now, the somites, they form these pairs. So we have 4 occipital, which make the brain, 8 cervical, which would make up the vertebrae, 12 thoracic, which make the thoracic, 5 sacral, and 5 to 7 coccygeal. So these turn into the spine and the axial muscles. Now, we've talked about this before. So the scleroderm, so this is the ventral and medial cells, they become vertebrae and ribs. Whereas the dermomyotome, which are the dorsal cells, right? That means the one close to the dorsum, to the outside here. So to the back, they become the dermis of the back, so it's the dorsal, therefore in the back, and the skeletal musculature. Now here's an alternate picture of what we were talking about. So this is the mesoderm before it becomes the somite. And what happens is that the various signals from the head down to the tail ensure that these unsegmented mesoderms become these segmented somites. So a good way to think about this is that these somites are just tissue that's been more compactly packed and more organized, and it's committed. So before it could have become anything, but now it's committed to become somite. And the somite develops as followingly. So this is the dermomyotome, and you have the different 2 layers. The scleroderm is beneath it, right? So it's beneath there. And we also have the dorsal aorta there. Now moving onwards, this is the development here. So we have the scleroderm, which is quite close up there. So that's the ventral cells, right? And we have the myotome, which are more dorsally present, same with the dermatome. So the cells migrate up, as we see here. And this is a properly sort of differentiated embryo where we can see a clear myotome. And this whole process here is called somitogenesis. And the whole reason for this is because you want to get mesoderm into somite and somites into scleroderm, dermatome, and myotome, which then become the different parts. So this whole process is just differentiation. Now it happens in a very interesting way. It's due to somitogenesis, that's the process. And this is quite a lengthy slide here. But somitogenesis, there's this clock and wavefront model. So what they're saying is that you have these gene expressions, so that's your WNT and your notch, so Wnt and notch, and they take turns on switching on and off in different cells. Now this sort of creates like a ticker, right? Like in a clock, you have a mechanism that sort of counts the time. So this creates like a clock. Now the wavefront is the molecular gradient created with notch signaling that moves craniocaudally. So many different proteins, this is your FGF proteins, your notch proteins, all of these things, they create a gradient. And at the top, so this is, think of this, the head, and this will be the tail, right? Excuse my simplistic drawing, but I think it's always good to see when this is simply explained. So at the top, we have a lot of notch. And at the bottom, we have very little. Now this gradient, right? What this gradient allows is the downward movement of the cells, alongside with the clock, which oscillates the gene expression, turning things on and off. And this sort of clock model, it comes from the neural tube. So what happens is that the mesoderm at the top, in the head, in the cranial region, accumulates notch. So it starts accumulating notch, right? And this induces the somite formation. And then the somite is formed. Once it's formed, the signaling subsides. So once this is formed, and the signaling moves down, and it moves down a gradient, and this is called the molecular gradient. Now we have also other proteins such as FGF and Wnt3a, which are the opposing proteins, which are more present in the tail, so they're more caudally and low in cranially, and this creates a gradient. And it's the different switching on and off between the two that enables the sort of movement of differentiation. So I hope that's quite the tricky part here. I hope that's I explained that clearly. Now then we have somite differentiation. So once we get the somites to form, and we get this primitive mesoderm, like these unorganized cells with a lot of potential, but very unorganized, and due to the notch signaling, they become somites. But now that we have somites, we need to sort of differentiate them. So the neural tube is the master signaler with the notochord. Their function is to sort of help the other tissue organize themselves. They organize themselves first, now the other tissues have to follow. So they produce WNT and BMP proteins, which activate muscle-specific transcription factors. And this is in the dermomyotome. So these are, so it's MyoD is one example for muscles, and YF as well. Now notochord produces something called sonic hedgehog. I know embryologists are quite crazy with their names, but it's called sonic hedgehog and noggin. It's called noggin because it starts in the head and goes down. So you can see they have a weird sense of humor. But this induces sclerotome formation. Now, I think I have a picture here. Yes, I do. This next picture here. So as you can see, the ones which are 1A, that starts to be the dermatome, 1B is the myotome, and 1C is the sclerotome. But these cells, they migrate. So the cells which are closest to, quite close to the notochord, this is your sclerotome, they receive their signaling from the notochord. Right? So it's just a proximity thing. Whereas the neural tube ones, the ones dermatome, myotome, they primarily receive it from here. And it's just because of the proximity. So these are some somite terms. This is a very useful diagram to know. I cannot stress enough how useful this is, because this came up in our exam and we had to label one of these, and everyone freaked out because it was a big question worth a lot of marks. So do not skip out on this. Now let's make some sense of this. So we have something called, so the myotome is divided into two parts. You will have the back region, so that's called the epixialis, and the ventrolateral region, which is the hypaxialis. So the back region, as you can see, is close to the back. So if you can imagine, this is the back, that's the front. And this here is the muscles, or the cells will differentiate into muscles which are spinal extensors. So these are muscles which will be spinal extensors, and they will basically form the support of the back. Now the ventrolateral muscles here, or the cells, they'll become spinal flexors. In the shoulder joint, they'll become shoulder muscles. In the hips, they'll become hip muscles. So in different parts of this, in different sections, I should say, in different sections. So here, that's the shoulder. Here, that's the hip. And here, where there's no really joints here, that's where they will become the different spinal flexor muscles. They will also become the trunk walls in this area, and extremity muscles once the limbs start budding. Okay, so that's the whole somite. That's like somite explained quite simply. And the reason for the somite, as I've said it before, is so that we can get the paraxial mesoderm to be differentiated, and so that the somite can form these units in the right locations. And once these locations are formed, they can transform into the relevant body parts. So that's either your spinal muscles, it's your shoulder muscles, or it's your skin and the connective tissue. So the whole point of these somites is to facilitate this along the neural tube. So moving on, myotome differentiation. So we have something called myoblasts. And myoblasts are these cells. They are myotome cells, and they lead to muscle cell precursors. So myoblasts, if you think to my video on bones and tendons, we had osteoblasts. So these are immature bone cells. Tenoblasts, immature tendon cells. Right. So if it's a blast, fibroblast, right, these are immature cells. But once you become a cyte, or once you become a myocyte, that means you're a committed cell. So we're not there yet, but they are forming there. And what we know about muscles is that once muscle cells form, they stay there. They can grow, but they generally don't tend to divide. Now this differentiation is created by something called myogenin. So myo means muscle, gen means generate. So it creates these muscles, or it enables this differentiation from the myotome cells, which are just cells that have been allocated in a specific region, to become committed muscle cells. And you have various growth factors that influence this. most key ones being myode, so MYOD, and MYF5, which are transcription factors. Now, once these growth factors are depleted, just like in the somites, so they depleted, the division stops, and the myoblast starts secreting ECM. And the ECM here, it's not collagen, like in tendons we have collagen tubules. It's not the case here. It's myotubules. So these are the muscle fibers which gives you your striations if it becomes skeletal muscle. And it's the contractile units. So if you remember back to the muscle video we did, it's these tubules which enable contraction. It's the units of contraction. So that's your myoblasts. Now we have a few other things. We have something called the lateral somatic frontier and the myotome domains. Now the lateral somatic frontier is just a location basically between the somites and the lateral plate mesoderm. Now if we can look at this photo here, it's right there. So that's where the sort of myotome or somite ends. So that's the somite right there. And that's the other lateral plate mesoderm. Now you have dermis. So the dermis in connective tissue forms both from the dermatome, from the somite, and from the lateral plate mesoderm. And this border just defines, this lateral somatic frontier defines the border between the two. It becomes more relevant in limb development. But yeah, and then we have something called myotome domain. So I found this very confusing initially, but it's primaxial and abaxial, which is different from epaxial and hypaxial. So primaxial refers to muscles that are primarily derived from the somatic mesoderm, basically the myotome. Whereas, so these muscles here will form muscles of the back. They will form erector spinae muscles, deep back muscles, right? So these are deep back muscles. And this is because they gain their signals from the neural tube. So it's going to be directly here. Whereas the abaxial muscles, right, they are from the lateral somatic frontier and lateral plate mesoderm. So they get their signal from the lateral aspect. And this becomes your infrahyoid muscles, the ones below your hyoid bone, your abdominal muscles, and eventually once you have buds for limbs, your limb muscles. And surprisingly, this also changes the innervation. So the muscles which are primaxial, right, which is your intercostals, your rhomboids, the lat dorsi, they are typically innervated by the dorsal rami of spinal nerves. And you know, this is a different part. Whereas in the muscles with the limbs, they're innervated by the ventral primary rami of spinal nerves. And we'll have a little slide about that later. It just shows that the nerve cells, which is not covered here, but they sort of come hand in hand with these muscle cells. So depending on the location, it also changes the innervation. So that's why you have like a brachial plexus, but it also innervates part of, innervates the shoulder and innervates part of the back as well, like the latissimus dorsi. So that's because of this embryonic remnant, right? So that's the innervation there. And we can get into all that, but that's just more complicated. I think this just keeps it simple. So we have primaxial, which is from the neural tube and notochord, abaxial, which are further apart and therefore receive signals from the lateral plate mesoderm. Primaxial, since they're quite close to the neural tube, which becomes the spinal cord, they become the muscles of the back and supporting muscles, whereas the abaxial muscles, they're further away, and these becomes your limbs, your organ wall muscle, and your limb muscles. So that's the separation between the two. So now we'll talk about the genes which regulate this. And there's many genes, right? You don't need to know all of them unless you want to become an embryologist or you want to study this. So I'm just going to focus on the key ones. So transcription factors, as we know, are required to activate specific muscle genes. So in order to activate these specific muscle genes, you need specific transcription factors. These are your MYOD, so MYOD, and MYF5. Now, MYF5 primarily enables differentiation into myoblasts, so that's its key function. Whereas MYOD or MYOD, it converts non-muscle cells into muscle cells. So this is very important. Now they've done experiments where they've bred animals and embryos where you had ones which were deficient in MYF or MYOD or both, and they didn't develop muscles. They didn't develop any muscles. So these transcription factors are key for the muscle development, and it's these two things. Now, if we move a little bit further, signaling, the notochord produces your sonic hedgehog and noggin. The lateral plate mesoderm produces Wnt and BMP, which activates your MyOD and MYF5. So in order to get these to be activated and cells to start expressing them, you need the outside influence, right? So from the neural tube, you need, and the notochord, you need BMP and WNT, and if it's further away, the lateral plate mesoderm supplements these instead of the neural tube. So depends on the location. So the abaxial muscles, right? Abaxial muscles, they get it from here, whereas the primaxial muscles, they get it from the neural tube. So the neural tube is for primaxial, lateral plates for abaxial. Wnt and BMP. Now the notochord produces sonic hedgehog and noggin. That's for sclerotome. So that we'll call that SCL. So that's for the sclerotome. That's not really relevant for muscles. But yeah, so they activate these muscle-specific genes and allows these cells to become committed muscle cells. So as I said before, mesoderm, sort of big picture overview. You have your mesoderm, which is primaxial, commits to becoming a somite, so they're packed together in the right location as the embryo goes. Somites become differentiated into sclerotome, dermatome, and myotome. And now myotome, you need to specifically find those cells to become myoblasts, which become muscle cells. And in order to do this, you need MyOD and MYF5. How do you get those? It's through signaling. So you need lateral plate mesoderm or neural tube, depending on the proximity of the towards the specific cells. That gives you Wnt and BMP, and that activates them. So I hope that's simple enough there. Okay, if you have any questions regarding this, do let me know. I know I'm going through it quite quickly, but I think it's quite simplified. And I think as long as you keep it, you don't even need to know the specifics, right? If, let's say this is a written question, you need to know the specifics. But if you keep in your mind that it's your mesoderm to somite, somite into differentiated, so dermat myotome and sclerotome, into myotome, and then myotome you need cells to commit to become muscle cells, and you do this through your myode and MY5. So next is the the innervation of the axial skeleton. So this is once you start building the limbs. This is the limb budding. So we'll cover this later in a future video, in the next week's video. But as you can see, the back muscles quite close to the notochord, they're called epaxial, whereas the hypaxial muscles are the ones further away. So that's the body wall muscles, that's the extensor muscles and flexor muscles. And basically what happens here is that each myotome receives innervation from the spinal nerves of the same segment. So you have a somite, so once again, head, tail, this is your somite, this is your somite. This is other somite, other somite. So the muscles at this level, right, they'll only be innervated by this level. The muscles at another somite level will only be innervated by these ones. Now, as you grow as an embryo, this all separates and then you elongate. So certain muscles might be in weird places. So a good example of that is the latissimus dorsi, which goes all the way down like this. Yet it is innervated by the upper section right here. And that's because embryologically it starts there before it migrates. So as we said before, the epaxial muscles, the ones in the back, get the dorsal rami, so that's this one here. It's the dorsal. Whereas the hypaxial muscles get the ventral ramus of the spinal cord and the spinal nerve. So the one that goes to the back, that's epaxial. one that goes front is for the hypaxial. And finally, this is just a sort of overview, just to sort of solidify the point. Depending on the area of origin, that's where you get the innervation, that's where you get the muscles. And you can see here there's a limb budding here in the lumbar region, which would be, in the thoracic region, which would be your thorax and your arms. And here you have your legs. So there's a pattern in this. It's controlled by signals. In the head, it's the sort of neural crest that does everything. In the cervical and occipital regions, it's the somite, so it's the somite in that specific region. And for the body wall and limbs, it's primarily the lateral plate mesoderm that contributes most of the signaling to develop. So yeah, we're going to move on to questions now. So this can be a question, this could be your MEQs. Outline how the myotome differentiates. Now, once again, you'll have to start from the beginning. So you'll have somitogenesis. So you say, how do the somites form? So that's your prime axial goes into differentiates into somites, and you have notch signaling, which goes craniocaudally. And your somites, they differentiate into your sclerotome, dermomyotome, which splits into dermatome and myotome. Myoblasts form from these myotome cells under the influence of myogenin, so that's the growth factor. And transcription factors MYOD5 and MYOD and MYF5 are required for this in order to allow these cells to become committed. Muscles and myoblasts are in the primaxial domain. They get signal from the neural tube and are not migratory because they're quite packed together. They become the muscles of the back and tissues of the back. Myoblasts, which are however found in the away from the neural tube and close to the neural plate, lateral plate, they receive signals from there. These are migratory and they become the body walls and the limbs, and they migrate where they need to. So this is just a little breakdown of that. Next sort of question we have is compare and contrast the development of skeletal, smooth, and heart muscle. So we've had this before in our muscle video that we did, where we compared the differences between the two. Now this is where they originate from. So the smooth muscle comes from the splanchnic mesoderm. So if you remember back, that's the mesoderm that's that sort of to the side, and it creates the splanchnic coulomb and the somatic, so it's below there. This is where it lines the insides of things. Smooth muscles is around the gut mostly rather than anywhere else. And its differentiation factor, this is purely for like MCQs, is serum response factor, so that's SRF. Now, basically all you need to know for this is that it's responsible for smooth muscle differentiation, and it does this through a signaling cascade by kinase phosphorylation pathways. And other growth factors help promote this and symbiose with this. So signaling pathway, that's the factor for smooth muscles. Now we move on to skeletal muscle. So that's the myoblasts, which fuse to form these multinucleated myotubes, which have the striations and the mitochondria. So this is myogenin, MyOD, and MYF5, which is your transcription factors. Tendons, which are sort of continuous with muscles, they are derived, however, from the sclerotome. So that's, as I said, connective tissue. And the transcription factor is sclerax. This one here. That's a good one to know for like MCQs. Cardiac muscles, origin, they are from the splanchnic mesoderm as well, but these are the ones which surround the heart tube. So the heart tube is a specific organ which starts to develop the heart in around week 4, and it's the cells, this is all proximity. It's the cells found near the heart tube that become cardiac muscles. So this means that they are striated. However, they have these intercalated discs that we talked about before, which makes sure that the myoblasts stay together. Now, differentiation, it's not MyoD. It's not involved in early cardiac development. It's something called Tin Man. And Tin Man is from Wizard of Oz. He didn't have a heart, so he was looking for a heart. And so this is why that's named after him. So it's Tin Man, which is responsible for cardiac muscle specification. And once again, to simplify this even further, it's where these tissues are found. All these cells are found in specific places. They receive signals from the tissues which are relevant to them. In the case of skeletal muscle, it's myogenin, MYD, MYF5. Cardiac muscle, it's Tin Man, and a smooth muscle, which is from the splanchnic mesoderm, it's SRF. So outline the molecular regulation of Somite differentiation. So we've been through this a little bit, but this is sort of like a good answer, clean answer for this. I would say that the neural tube is the master signaler. WNT and BMP proteins activate MyoD in the dermomyotome. Under neurotrophin, the dermatome becomes the dermis. So that's a new one here, neurotrophin 3. And these factors, MyoD, MYF5, are required for myoblasts to proliferate and enable the conversion of non-muscle cells to muscle cells. Finally, the notochord produces sonic hedgehog and noggin to new sclerotome. And for bonus points, you could even say that the lateral plate mesoderm supplies BMP as well, to ensure and went to ensure differentiation of the the primaxial myoblasts. So that's for bonus marks. And once again, this is a good diagram to have. So it's the location of the cells, so the notochord, sonic hedgehog, that's SHH, BMP4 from the neural crest cells, and the neural tube went into the muscles here. MYF5 and MyoD are required for both muscles to work. And then you also have neurotrophin 3 somewhere here. Yep, neurotrophin 3, which makes the dermis. So this is quite a good picture to sort of memorize in your head. So this is a very common question. It's label the following structures. Ignore this 3 here, that's a duplicate. So number 1, this is the lateral plate mesoderm because it's on the lateral side and it's on the top. Number 2, this is the intermediate mesoderm because it's in between. Number 3, this is your somitic mesoderm or your primaxial mesoderm. But once it becomes like this little thickening here, that becomes somitic mesoderm. Number 4 is the notochord. Number 5 here, that's the endoderm. This here is your ectoderm. And number 6, that's your neural tube. So there we go. Once again, it's just a different version of this diagram right here. So you have your somitic, you'll have your intermediate, and then you will also have your lateral mesoderm right there. So somitic, intermediate, lateral. Now this picture, this is what we got in our exam, was a big blurry microscopic picture. It was like this. It was not labeled, and we were asked to label all the drawings, like as many things as possible, and it was like 12 marks. And so what you would start with is that you would label this. This is the ectoderm here, that outer region. This is your somatic mesoderm right there. We have the endoderm. This is called the dorsal artery, so this is the artery at the back. Basically, it's just learning this here, this picture. So A would be the neural tube. So this is an incompletely closed neural tube, which will eventually close. B is the notochord. So once you find the neural tube and notochord, you're set. Therefore, what's adjacent to it? C, it must be part of the somites. But in this case, these are already differentiated. And how do we know they're differentiated? It's due to this angle here. You see how this follows the angle? So this is about to close up. So this is how we know it's more differentiated. So in the bottom region, that becomes the sclerotome. Whereas on the top region here, that would be our dermatome. D would be dermatome. And E right underneath would be the myotome. Now eventually what happens is that the sclerotome moves up here like this. and this gets reorganized, and this sort of moves to the bottom like that. But we're not there yet, and that's why it's quite tricky. You would probably get marks if you call this a somite, but they want the specifics, and they would be referring to this picture right here, where the dorsal medial cells, you have the dermatome, you have the muscle cells just beneath it, and you have the sclerotome. Another picture. So we have to break this up. Then this is a drawn picture. You could have this as well. So it's like a hand-drawn picture. Once again, it's the same thing. So we have to identify our neural tube. We've identified our notochord. We found our ectoderm. We found our endoderm. And then we can go to the rest. So, as I said before, this is your coulomb. Number 4 is your coulomb. That's the lining or the inside of your gut. 3 will be your parietal layer of the lateral mesoderm, also known as the somatic layer of the lateral mesoderm. This would be the splanchnic or the visceral layer, the one that's inside the organs, or close to the organs. 4 with the coulomb in between. Number 5 is the notochord. Number 8 here is the neural groove. So the inside is the neural groove, outside is the neural tube. And then we have these things here. So we're looking at number 1. So all of this here was once a somite. So this all together was once a somite. It was just a collection of cells. But now they've sort of differentiated. And in the back here, more sort of more ventrally, here, because this is the ventral aspect, that's the dorsal. So a little bit more ventrally, we have our sclerotomes. These are sclerotome cells, like little dots. Then we have 1B, which would be our myotome, and 1C, which would be the dermatome. Once again, it's the same picture here. And that's what the things are there. And we have the ectoderm, neural groove, and dorsal artery. I forgot number 9. That's the dorsal artery. Now, this is just a drawing to sort of show the changes that these things undergo. So once your neural tube sort of closes up, as we see here, all these other tissues move downwards. So we have our somite here. We have our intermediate mesoderm. We have all that. So that's our intermediate mesoderm. And then we have our lateral mesoderm here. So that's somite, intermediate, lateral. But what we see here is that within it, we start to see some cells differentiating. And what happens is that this expands, this sort of uncoils outwards. And the top part becomes the dermatome, middle part is the myotome, and the most ventral part would be the sclerotome. Now that's the end of the slides there. I hope this has been useful. I know this is a very tricky topic. I know a lot of people do not like this topic, and a lot of people skip it. But it's quite important when you try to understand the anatomy and the innervation. It's very important if you want to do surgery, so embryology is quite important, especially if you want to do pediatrics. So embryology, you really need to have a good grasp on that. This was the first part, so this was just muscle development rather than anything else. The limb development and the bone formation will be covered in next week's video, because otherwise the video would have been too long and it would be too difficult to sort of understand everything in sort of one piece. And once again, once we understand the muscle development concepts, we can apply that further for the limb development. So if anyone has any questions right now, please let me know. So someone is asking about Tin Man. Yeah, so Tin Man has it has been discovered in fruit flies initially. And if they do ask you about the sort of gene or transcription factor that is required for cardiac muscles, I would say Tin Man. That's always a good bet, because especially in an MCQ, they might ask that. Because in MCQs, even in the embryos they show us here, it's not human embryos that you'll be shown, like this one here. This is from a rat, I think, I'm not sure. But they use sheep embryos, they use rat embryos, they use chick embryos. So just because it's not a human embryo, or the knowledge is not about a human one, does not mean that you should ignore it. But yeah, that's a good question. So yeah, Tin Man, it was discovered in flies first, but if they do ask about it in the exam as an MCQ, I would definitely put Tin Man. Basically, in humans, it will be some other factor. It's just that it's more difficult to isolate. And the way the people isolate these different cells is because they express different markers. So if they, for example, express transcription factor MIOD and MIF5, then that's how you know that cell is becoming a committed muscle. It's going to become a committed muscle cell. It's because what these cells express. Any other questions? Does anyone want me to go over something again? I know I went quite quickly through the genes and the regulation, like the notch signaling right here. No more questions. Right, someone's typing something up in the drive. No. I mean, if there are no further questions, well, you can just keep. Oh, there's another one. Can you possibly go over the difference between prime axial, abaxial, epaxial? Okay. So regarding Tin Man, it came. I think it came up in second year. I don't remember, but I do remember it coming up in exam. That's why I thought I'd just include it. And you never really know with the examiners. Another question is: could you possibly go over the differences between prime axial, abaxial, epaxial, and hypaxial? Okay. So all of these terminologies are. So this is, I think, a good diagram here. All of these terminologies are relative to the muscles. So it's epaxial and hypaxial once they become actual muscles. And that just means they're relative to the axial skeleton. So if we think of the axis there, right, that's our skeleton. And these are our muscles. So this becomes above, sorry, I just back again. Any muscles which are here become epaxial, which means above the axial line, whereas the ones become below are hypaxial because they're below. So this is once we have muscle development. This is like once weak force strikes. The prime axial and abaxial, so these are domains of they're domains of the tissues. So what this means is that the prime axial domain is referring to the mesoderm, right? It's referring to the myotome before it becomes a muscle. Whereas once you have muscles forming, that's when epaxial and hypaxial start to matter. So it's just a different terminology at the different points in time. So prime axial, think mesoderm, right? Very early on. And a prime axial, abaxial. Whereas once you have muscles forming, once the embryo starts looking like, starts forming muscles, starts becoming more specialized, that's when you have epaxial and hypaxial. Another way to think about it is epaxial and hypaxial is when you start having limb development, whereas prime axial and abaxial is just when it's cells floating around. I'm trying to find the slide here. Yeah, myotome domains. So as you can see, this is much simpler. The embryo here is way simpler. And the ones which are sort of close to this area, so this would be your prime axial domain, whereas this would be your abaxial domain. And it just refers to the signaling. and the cell origin. So lateral plate, mesoderm is abaxial, and primaxial is your notochord and neural tube. Oh, the abaxial, I just found it confusing with the different myotomes. So hypaxial and epaxial, and then sometimes they will talk about primaxial and abaxial. I find it quite confusing when it was an embryo. What is the front, what is the back? It's not always clearly explained. So if you think about this as the back and that as the front, I think that might help with visualization of the embryo. So this is your front, this is your back. And it's just a way to describe the different positions, the different anatomy within the embryo and the different positions. Just how like we have, anterior and posterior. This is the same thing. It's just terminology. But yeah, like here we have epaxial and hypaxial. It's just the terminology here. And different terminologies used for different things. But in regards to the myotome specifically, it talks about the different signals. So sometimes you get signals from the neural tube and the notochord. These are the ones which are closer to the neural tube, and the ones which are further away, which become your limbs, these get from the lateral plate mesoderm. So I hope that sort of explained the difference there. Yeah, if any other questions, just write them down. Or if you're rewatching this video, and just put them in the comments, and I'll get back to you and I'll answer them. So someone has just, I'm just looking here, someone has just asked if we need to know about the different deformities in muscle development. So the deformities become more relevant once you get limbs, because if you have failure, like let's say in the MyOD and MYF, if they fail to sort of proliferate and fail to sort of go through, then you'll have no skeletal muscles at all. Now, as you can imagine, that may be compatible with life, but the embryo will not develop fully to term. Basically, what will happen. So once you have any muscle problems, these muscles will be needed for breathing. These muscles will be needed for a lot of different functions. If they do not become developed, then you won't have a compatible with life outcome. But yeah, we'll talk about different development problems, which I think you're referring is what you're trying to imply, in the next week's video, where we talk about limb growth and what happens when it goes bad, or what happens when there's certain exposure to different types of chemicals during the pregnancy. Yeah. Any further questions? For those still watching, I'm just curious. Do you find this helpful? Was this a helpful way to do embryology? Would you like more questions? Because I'll be honest, I was quite struggling to make the this presentation for this week. because there's just so much to cover and it's quite complicated. It's different concepts coming together. So I was just thinking, is it good to split it up like this? Would you like a video on life before birth? Would that be helpful? Maybe about the neural tube development, maybe that might be of use. Or is this the right level of detail, or does this feel too detailed? I'll be sending out a feedback form, so do please fill it out. Especially if you want the copy of the slides, we'll be sending out a feedback form that you can fill in. But yeah, I hope that was helpful. It's a very tricky topic, and yeah, best of luck. And if you liked it, like the video. If you have any questions, comment, subscribe, all that stuff. And yeah, thank you for tuning in. Have a nice day.